us has been his home for a long time. And now, as also from the past, he has been doing different projects about pollination and other entomological work in different agricultural landscapes, and is now going to tell us about reed pollinators and honeybee management for crop pollination. You are welcome to talk to us about this very important seminar today, and just feel at home. And I would like to wish you all a good seminar today.
Super.
Over to you, Maxime.
Thanks. I'll now share my screen. Normally, you should see the presentation.
Yes, I
I
see.
think. Super. So thanks for the introduction. As introduced, I'm Maxime. I'm a postdoctoral researcher at the lab of Professor Chris Verheyen, the Forest and Nature Lab it is called, at Ghent University in Belgium. And I'll present to you the talk titled Wild Pollinator and Honeybee Management for Crop Pollination. And first a bit about myself, about my academic career so far. So from 2016 to 2019, I conducted my PhD research at Ghent University at the lab of Professor Guy Smagge and Dr. Ivan Meus. Then for 9 months of 2021, I went to Washington State University for my first postdoc at the lab of Professor Lisa de Vetter, where I mainly focused on optimizing the use of honeybees for pollination of blueberry. My PhD, on the other hand, more focused on how landscapes influence the pollinator populations in sweet cherry orchards and apple orchards, and how that the landscape and these pollinator populations, both wild pollinators and honeybees, then influence sweet cherry pollination. After my time in Washington, I went to Michigan State University at the lab of Professor Rufus Isaacs for 1 year to focus on more synthesizing the research instead of doing field work, writing reviews and meta-analysis on pollination of fruit crops, both with honeybees and wild bees. And in November 2022, I transitioned back to Ghent University, but at a different lab, so at the lab of Professor Chris Verheyen. And most of my work, my research, has focused on pollination, and pollination is the facilitation of fertilization of flowers, whereby these flowers then develop seeds and fruits or both. And pollination, or this fertilization, is facilitated mainly by insects, because as insects are at least in my study systems, insects are the most important pollination agents. And while insects are visiting flowers for nectar and pollen, they transfer pollen from flower to flower, and therefore they accidentally then also pollinate these flowers. So as I mentioned, insects are the main pollinators in the study systems that I've worked on so far. And for instance, as an example, in Belgium, we have one species of honeybee, Apis mellifera, and then we have a lot of wild bees, which are the bumblebees and the solitary bees. And for the bumblebees, we have around 25 to 30 different species. Compared to the solitary bees, which is a very, very diverse group. We have in Belgium, which is a very small country, around 400 different species. And then we also have a lot of other pollinating insects like hoverflies, butterflies, beetles, etc. But mainly the honeybee and the wild bees. So the bees are the main pollinating insects over here, because bees, the larvae of the bees are fed 100% with pollen and nectar. So these bees, they forage very intensively on flowers to collect these resources, while the other pollinators like the hoverfly and butterflies, their larvae, so their offspring, is fed with many different other substrates. So they forage less intensively on flowers in general. For the remainder of the presentation, and also for the European and the Northern American context, it's important to keep in mind that the honeybee is a very, very different animal compared to the wild pollinating insects, because the honeybee actually in our regions, it is it does not exist in the wild anymore. The honeybees, they are kept in boxes, and they are tended and cared for by beekeepers. And if these beekeepers do not care for their bees, then these colonies don't survive the winter. Compared to the bumblebees and the solitary bees and the other pollinators, these pollinators are wild species, and they maintain their populations by themselves based on the resources that are provided in the landscape. We also know that pollinating insects are in decline, and this is caused by multiple interacting drivers. As you can see on the figure, pollinating insects are under pressure due to habitat loss, loss of floral resources, pesticides, pathogens, also non-native pollinators, etc. And this figure is a very complex figure, but I mainly chose this figure to indicate that all these different drivers and these pressures are interacting with each other. However, we do know that the loss of habitat and the loss of floral resources is the main, one of the main pressures or drivers of pollinator decline. And this is mainly caused by increasing area of intensive agriculture. Another interesting thing to introduce is that during the last 2 decades, there have been a lot of studies on pollination of crops. So these are observational, site-replicated studies that look at the gradient of pollinator visitation of both honeybees and wild bees and their effect on crop pollination rates. And 2 big meta-analyses, these ones, so Garibaldi et al. And Danese et al., these 2 big meta-analyses have combined a lot of the data of these pollination studies across the world, and they have looked for general patterns. And the main conclusion, one of the main conclusions of these meta-analysis is that there's a considerable positive pollination contribution of wild pollinators. So as you can see on the x-axis, you see pollinator richness, or this could also be pollinator abundance. So if you have more wild pollinators that visit the flowers of your crop in your field, you will have higher pollination rates. So crop pollination clearly increases with wild pollinators in multiple crops across the world. This pattern, however, was not detected for increasing or decreasing honeybee visitation. So based on these 2 meta-analyses, a lot of people often have the idea that wild pollinators are essential for crop pollination, and honeybees are less important. However, there are also other studies that conclude that there's a considerable pollination contribution of managed honeybees. For instance, these are mainly the studies that calculate the economic added value of both honeybees and wild bees. And for instance, the study of Klein et al. From 2015 calculated the value of all crops globally of both honeybees and wild bees, and they found that on average, both honeybees and wild bees at a value of $3,000 per hectare for agricultural yield. Another study is that of Riley et al., which focuses on crops in the US. And they calculated this monetary value for various crops for both honeybees and wild bees. And they found that for both honeybees and wild bees, it's around 2 billion that these species add to crop yield. And they also, so Riley et al. Also focused just on almonds, and here they found that honeybees add a value of 4.2 billion to the agricultural yield of almonds, whereas wild bees, in the case of wild bees, it's almost zero. This, of course, can be explained by the fact that almonds in the US are big monocultures. There's no habitat, so there are no wild bees, so they can also not contribute. While the honeybees are all the honeybees in Northern America, more or less all the honeybees, are put into California almonds during almond bloom, which is around February. So therefore, the honeybees have a massive impact on almond yield. Another interesting point to make or to present is the cultivated area of crops worldwide. So the study of Eisen et al. From 2019 investigated the trend of, or the increase of the area of crops, or the cultivated area throughout the last 50, 60 years. And they have looked at the percentage of change, as you can see on the y-axis, and the years in the x-axis, and you clearly see that the area of crops worldwide has been steadily increasing throughout the last decades. And they also have looked at the area of crops that depend on insect pollinators. And here you can clearly see, so the yellow line represents the area of crops that depend on insect pollinators, that this increase of the area of cultivation of insect pollinator dependent crops during the last 20, 30 years has increased more pronounced. And from this, we conclude that there's an increasing dependence on pollinating insects in agriculture. And if we now look back on what I've introduced so far, we have seen that both honeybees and wild bees are very important for crop pollination. But we also know that these pollinating insects are in decline. But on the other hand, we see that there's an increasing dependence on pollinating insects in agriculture. So this is contradictory and might cause a problem. So this has brought to life the concept of integrated crop pollination, and this concept aims to safeguard crop pollination. And we aim to do this by integrating different pollinators, so managed honeybees, the wild bees, but also alternative managed bees, so a couple species of wild bees you can actually also manage like honeybees. And we aim to integrate these pollinators with a series of practices, agricultural practices like pesticide stewardship, horticultural practices, and habitat enhancements. So within the concept of integrated crop pollination, we aim to develop evidence-based and targeted measures that benefit insect pollinators, their pollination contribution, and therefore the yield of the crop. And for the talk of today, I will mainly focus on the three different ways to manage bees. So the wild pollinators, also alternative managed bees, and the honeybees. And I will, for each of those topics, highlight some studies that are relevant to advance the current knowledge in order to improve management of these different types of bees. So let's start with the wild pollinators. Regarding the wild pollinators, we already, during the last 2 decades, this has been studied very well and very extensively. And for these wild pollinators, we know very well which parameters determine their pollination contribution and whether or not these populations are healthy or not. For instance, we know that landscape composition is very important. And here, so landscape composition is the composition of the landscape indicates a certain amount of a certain habitat type. And here we know that more semi-natural habitat or more natural habitat is better for having healthy pollinator populations that also visit your crop. On the other hand, we also have landscape diversity or landscape configuration. And here we know that crop diversity, field size, and edge vegetation all have a very important and relevant impact on pollinator populations. And we also know that field scale, a lot of field scale measures like planting flower strips, um, extensive weed management, planting hedgerows also has a positive effect on wild pollinators. So while we know a lot of these, it has been very well studied. The fact that we know that certain parameters and habitat types are important does not always. It's not always easy to translate it to management recommendations for stakeholders and policymakers. Definitely, for instance, semi-natural habitat. How can we manage this? We know for sure that more natural habitat is better, and it's also often studied with linear models, so linear regressions. So if you find a positive relationship between natural habitat and wild pollinators, it's a linear relationship, and this then leads to the recommendation, the possible recommendation that more natural habitat is better. But this is difficult for farmers and policymakers to act upon because you don't know where to plant more habitat, and you also don't know how much natural habitat do you need. So this brought me to the question regarding the natural habitat, how much semi-natural habitat is actually needed. And 2 years ago, there was a very interesting policy perspective, so a kind of review paper by Lucas Garibaldi et al. That suggested that working landscapes, so agricultural landscapes, need at least 20% native habitat. And this really sparked my interest to test this suggestion with empirical data. And so I did that. And how did I do that? I tested 3 different models with empirical data. And the first model I put forward is a null model. So this implies that with more semi-natural habitat, you have no effect on wild pollinator visitation. So there's no relationship between those 2 variables. Then you have the linear model, which indicates there's a positive relationship between natural habitat and wild pollinator visitation. And this linear model is the most commonly applied relationship for modeling these 2 variables. And what I then did is also added a nonlinear model. So this nonlinear model suggests that there's a positive relationship between semi-natural habitat and wild pollinator visitation up to a certain threshold, beyond which more natural habitat does not yield more wild pollinators in your fields. So this nonlinear model would, with this nonlinear model, we would be able to establish a certain threshold, which would lead to a more quantitative policy recommendation that can be used for mitigation measures. So I then analyzed some data that I collected during 3 years of my PhD. So in 2015, 2016, and 2017, I collected pollinator data in Belgium, so here in the center of Europe. The dots on the map represent the different orchards, and the different colors represent the year in which it was sampled. So in total, I sampled pollinators in 25 orchards during cherry bloom. And we then also mapped the landscape around each cherry orchard. And then with this data set, I tested this null model, this linear model, and this nonlinear model. And for these 3 models, I then selected which model best predicts or best explains the detected variation in the data. And the result is that the nonlinear model was the best explanatory model for explaining the variation in the data. As you can see here, with increasing semi-natural habitat, wild pollinator visitation increases up to a certain point, beyond which more natural habitat does not imply that wild pollinator visitation increases even more. So with this nonlinear model, you can also test. You can also test. Yeah, this is the laser. You can also establish a threshold, and I found that this threshold is 15% of semi-natural habitat, and this leads to a quantitative guideline versus the use of linear models, which just say no more data, more natural habitat is better. And this also implies that for 70% of the orchards, it would be beneficial to increase habitat. And we then also know which orchards we need to focus on in order when we want to add or create natural habitat. So yeah, I wanted to present this as it's a small study, but it's an interesting concept that builds on the gigantic body of research on wild pollinators. So then over to the alternative managed bees and how to manage these species. So it's called trap nesting bee management because we have some solitary bees that can be managed with artificial trap nests, like mason bees and leafcutter bees, and each different parts of the world have different species that can be managed. Um, and these solitary bees, they don't live in a colony with a queen like bumblebees or honeybees, but with these solitary bees, every female is making her own nest, and this is something we can study with these artificial trap nests made out of bamboo and wooden groove boards. And within these cavities, they build their nests. So they build multiple brood cells, and in each brood cell, they provide a food provision, an egg, and the egg hatches. The larva then eats the food provision and spins a cocoon. And after winter incubation, the bee can emerge the next year to uh, complete the next cycle. So in our study region, we worked with Osmia cornuta. It's this bee species. It's also called the European orchard bee. It it makes nests in artificial trap nests very easily. Um, the activity period of this bee species also overlaps completely with pear, cherry, so both sweet cherry, sour cherry, and apple. And we also know from pollen identification of pollen samples of this bee species that it forages intensively on pear, cherry, and apple. And from pollination studies, we also know that compared to honeybees and bumblebees and also hoverflies, this is a very efficient pollinator of tree fruit crops. Um, so therefore, we chose to work with this species. What we also know that growers and other people who rear these bees, they use a lot of diverse nesting materials. As you can see on the picture, bamboo, wooden groove boards, paper straws, cardboard tubes, drilled holes in wooden blocks. And everyone who uses a certain material is convinced that this is the best material for certain reasons. However, there is also the question to be asked, which is the material that the bee actually prefers? And there is not a lot of there are not a lot of studies that look at this with definitely not with site replication. So this might be an interesting question to ask. And we also know that there are a lot of parasites that are associated with these nests, like kleptoparasitic mites and kleptoparasitic drosophilid flies. And these kleptoparasites, they feed themselves with the food of the bee larvae. On the other hand, you also have brood parasites like brood parasitic wasps and brood parasitic flies. And these brood parasites, they feed themselves with the bee larvae itself. And both the kleptoparasites and the brood parasites are a major concern for growers and people who rear these bees. because we want to control these parasites and avoid that infestation of these parasites becomes too high. So this brings us to the questions of this specific study about trap nesting bees. The first one, which type of nesting material is preferred by the bees to make nests in? The second question is, how does nesting material, time in the season, and landscape influence bee development? And the third question was, how does nesting material, time of the season, and landscape influence parasite infestation? So in order to test this, we set up again in Belgium a study in sweet cherry. We selected 12 sweet cherry orchards. In each orchard, we then put a trap nest with 40 wooden cavities and 40 paper straws to compare wooden cavities with paper straws as nesting material. As you can see on the picture in the right, this is how it looked in the field, in the nests. So the wooden cavities were just wooden cavities, while the paper straws were just inserted in the wooden cavities. And then every nest was also provided with 100 cocoons of Osmia cornuta in order to make sure that we had bee activity at each site, so we could collect data. Then during 3 moments in time, we took photographs of the front of each trap nest. So during at the start of cherry bloom, during the middle of cherry bloom, and at the end of cherry bloom, we took a photograph. And with these photographs, we can also we can yeah we can determine when a certain when a certain cavity was filled, and where it was filled in time. So with these photographs, we can establish 4 time periods, and we then sampled 8 sealed nesting cavities per time period per nesting material. And from these 8 cavities per period and per nesting material, we opened them up in at the end of summer, and we counted the number of brood cells. We also determined the number of well-developed cocoons in the brood cells, and we also determined the number of brood cells infested with mites and the number of brood cells infested with Drosophilid flies, so the 2 parasites. Then the well-developed cocoons were also incubated. They were subjected to a winter incubation and a spring incubation, and after the spring incubation, we can identify the emerged bees, and we can determine if they are male or female, and thereby we can determine the sex ratio of the offspring. And lastly, we also conducted a landscape characterization. So we mapped the amount of semi-natural habitat in a radius of 500 meters of the trap nest. So as you can see here, in the center is a trap nest, this is the orchard, and these green polygons are the natural elements like forest, shrub vegetation, hedgerows, extensively managed grassland. So then to the results of this study. The first question was, which nesting material is preferred? So we calculated a nesting preference index based on the photographs to see which material most cavities were filled or sealed. And we found that the nesting preference index was a lot higher for the paper straws compared to the wooden cavities. And we found that there are 35% more nests made in the paper straws. And definitely in the beginning, the difference is a lot higher. So they have definitely an initial preference for these paper straws compared to the wooden cavities. The second question, how does nesting material, time in the season, and landscape influence bee development? Here we found that successful bee cocoon development was slightly higher in the paper straws compared to the wooden cavities. We also found that successful cocoon development decreases with time throughout the nest construction period of the mother bee. So this indicates that the fecundity or the fertility of the mother bee decreases towards the end of her life. Then we also found that the sex ratio, so the proportion of female bees in the offspring decreases slightly towards the end of the nesting cycle. And we also found that the landscape, so the percentage of semi-natural habitat, has a positive effect on the sex ratio. So you find 10% more female bees with increasing amount of semi-natural habitat around the nests. And this is also very important because the female bees in the offspring are the bees that will pollinate your that will provide the pollination next year. So if you have more female bees in your offspring, that's better for the pollination next year. Then the third question, how does time in the season, nesting material and landscape influence parasite infestation? Here we found that mite infestation was a lot higher in the wooden cavities compared to the paper straws. We found that there's 92% less mites in the brood cells in the paper straws. So that's also a very big difference. And then to conclude, we also find that mite infestation and Drosophilid fly infestation increase throughout the nest construction period of the mother bee. So throughout the season, the brood cells that are formed later have a higher rate of parasite infestation. So to conclude, we found that the nesting preference for paper straws, that the bees prefer to make nests in paper straws. We also found that successful bee development was higher in the paper straws and that mite infestation was lower in paper straws. And this can be explained, and this leads to the recommendation to use paper straws or similar materials that are completely round, like bamboo, for instance, instead of wooden nests that always have tiny cracks, and through shrinking and expansion of the wood, there will be holes whereby the mites can disperse and whereby you have increased humidity or increased dryness, which is not good for bee development. Second, we found that the sex ratio, so the proportion of female bees for next year, increases with natural habitat. So this leads to the recommendation. We can explain this because more abundant and more diverse floral resources support the higher investment in female offspring, because females, in solitary bees, females are a bit bigger than the males. So the mother bee needs, if she chooses to make a female, she needs to collect more resources, and she makes this choice more often in landscapes with more natural habitat because there are more and abundant and diverse resources. So this leads to the recommendation to make sure that you have sufficient natural habitat and resources around your orchard. And we also found that the sex ratio and successful bee development, successful cocoon development decreases with time throughout the life cycle of the mother bee, and that mite and Drosophilid fly infestation increases with time. So this leads to the recommendation to remove or to seal the nests after flowering of the tree fruit crops, because at that time the brood cells that contain most of the females and fewest of the parasites have been produced already. So after tree fruit flowering, you mainly have male bees and parasites, which you don't want. You don't want too many male bees because you already have more male bees than female bees. So that is it regarding the alternative managed bees, and I will now present a bit more on honeybees. And as I mentioned, regarding honeybees, regarding wild bees, we have 2 decades of a lot of research. For honeybees, however, it is a bit different. We know that farmer recommendations, so if extension services provide guidelines for farmers to recommend how many beehives or honeybee hives they should stock. These recommendations are at the field scale. but we don't know if this is actually effective. And I'm making an overview of field scale colony density and its effect on honeybee abundance in the field. And you clearly see there, it's still, I still need to do a small meta-analysis, but you clearly see no effect, no effect, no effect. A lot of studies point that there's no effect. And if there is a significant positive effect, it is because these studies have looked at fields with and without honeybee hives, and that's quite obvious. But if they look at field level hive density gradients, so more or less hives, most of the studies don't find an effect. In addition to colony density, we also don't know if colony strength, what the effect is of colony strength. So if you have bigger colonies compared to smaller colonies, is it better to have 2 or 3 big colonies compared to 4 or 5 small colonies? And then an important aspect as well is at what scale do we measure this? So the field scale or the landscape scale. So when it comes to honeybee management, there are still a lot of questions to be solved when it comes to managing these species for crop pollination. Compared to the wild bees, during the last 2 decades, this has been studied a lot less. So this brings us to the main question we wanted to study in Washington in 2021. Mainly, is it more appropriate to manage honeybee densities on the field scale, like you see on the left, or should we incorporate the hives in the landscape scale as well? And should we recommend growers to communicate better with each other on how much hives they should put in their fields? So in order to answer the question of field scale versus landscape scale, we conducted a study in Washington State in the United States, and we selected 16 high bush blueberry fields, and around each fields we mapped the landscape again. So here we determined the amount of blueberry fields, so the blue polygons, and we calculated the percentage of blueberry in a buffer zone of 250 meter, 500 meter, 750 meter, and 1000 meter around the central study site. Then during mid-bloom of blueberry, when the fields are fully stocked with honeybees, we mapped all honeybee hives in all fields. So both in the field of study, but also in all neighboring fields in a buffer zone of 1000 meter, we visited every field and we checked the parameters, and we mapped all hives, and we located the hives, and we counted how many hives are in each location, also in these alleyways, to get an idea like how many hives are in each landscape and at what distance. Then we also conducted transect walks. So during mid-bloom of blueberry, we caught all flower visiting honeybees for 2 hours. So that's always done in the central field. And these are some general results. So the field level hive density, so the hive density of the central field, we found variations of 3.5 to 25 hives per hectare with an average of 12 hives per hectare. And these are the units that the farmers manage. So we clearly see that there's a huge variation of how many hives each farmer put in the field. When we look at the landscape level hive number, in total, in the 16 landscapes, the 1 kilometer buffer zone, we mapped a total of 6,340 hives. Again, with a great variation of 16 to 920 hives per landscape, with an average of around 400. So again, a lot of variation from landscape to landscape. And the same goes for the honeybee count. So a total of 1,055 honeybees were caught on blueberry in the different fields. Again, with a big variation from field to field, 44 to 114 with an average of 74. Another interesting result is that we found a strong correlation between the number of hives in the landscape and the amount of blueberry fields in each landscape. Definitely at a landscape scale of 500, 750, and 1000 meters, we find strong positive correlations between the landscape and the amount of hives in the landscape. As you can see here, for the 1 kilometer radius, with more percentages of blueberry or more blueberry fields in your landscapes, the landscape level number of hives increases. And this, of course, is not per se surprisingly, and it's a finding that's very intuitively, but this is the first time that this correlation has been established in for a crop. And then the main result of this study is that we did a lot of statistical analysis, and we found that the landscape level hive number in the 1 kilometer radius is the most important factor predicting the abundance of honeybees on blueberry. And also we found that the field level hive density was not included in the best model to explain honeybee abundance. Um, and of course, this is an interesting finding because farmer guidelines are field level densities, but they don't explain the variation that we detected because they miss the contribution of the hives that are present in the wider landscape. So this brings us to the end of the talk, and I will summarize some take-home messages. The first one being that both honeybees and wild bees contribute to crop pollination, but this is very context specific. Like for instance, in almond in the US, it's all honeybees, while in cherry in Belgium, it's more wild bees, and in blueberry in Michigan, it is more a matter of both honeybees and wild bees. So it's very context specific, but we know on a global scale, both wild bees and honeybees are important. We also found that nonlinear models can be used to determine the threshold amount of natural habitat, which is useful for policy recommendations and also for planning mitigation measures in order to improve pollinator health and crop pollination. We also found that nesting material, landscape, and time in the season affect efficient management of trap nesting bees. And we find that honeybee visitation is determined by landscape level hives, and while field level recommendations miss these contributions. And these are the main studies that I presented in the 3 parts. All are quite recent, so from this year and last year. And with this, I would also like to thank all my collaborators, my supervisors, a lot of students and colleagues and my funding agencies at the different institutes that have helped me conduct this research. And if you have questions, this is my email address, so you can contact me. So yeah, that was it.
Wow, thank you very much, Maxime. That's a
Yeah,
great talk.
yeah, thank you.
Yeah, a cross-cutting research from agriculture, biodiversity, and also cash, livelihood improvement.
Yeah,
Yeah,
yeah.
I
In
would
order
like,
to have farmers on board, it needs to be relevant for their yield.
of course,
Yeah,
yeah. Actually, it is like beekeeping is beneficial in many angles. Yeah,
yeah,
and the new thing I got from this talk is that honeybee are only kept uh, in like beekeepers' hives, not in the wild. That was new to me. I was thinking maybe they also live in forests or in the other wild areas, but that's a very new thing, which means beekeeping is then more important than I was thinking.
Yeah, it probably is different. I think, for instance, in Australia, you still have a lot of wild honeybees. I think
Mm-hmm.
possibly in Rwanda as well.
Mm-hmm.
I once went to Belarus, and over there they also have both. So they have the wild colonies, but they also have managed colonies. But in Western societies, yeah, we just don't have the habitat anymore. Um,
Okay, yeah. Maybe Andrew, if he knows about that, he can tell us the status in Rwanda. If we have both the wild
mhm.
honeybees and the domesticated one. Yeah, I'd like to call upon everyone who may have a question or a comment to just write in the chat room or raise the hand or just unmute and speak. Yeah, Jaime, you are welcome.
Thank you so much. Thank you for the nice presentation. I've been following carefully because this has much common with my PhD research. Um, yeah, but Maxime, I was just very curious to know about this threshold of the natural habitat in the landscape that is required. Uh, like for example, if you what do you think is the type of this natural habitat? Because with the variation of the floristic diversity, like in the uh, broadleaf species, for example, kind of tree forest or just woodlots, and then also the nidro species. You see that the flower diversity is different, and then this can influence the abundance of the wild pollinators. So, what do you think about that regarding to this threshold of the
Yeah,
natural?
yeah, it's a good question, and it's um, yeah. What I did in that study was just combine all natural habitat types. So deciduous forest, mixed forests, shrublands, field margins, hedgerows, etc., and grasslands. And you clearly see here in this cloud that there is, it's not really clear what's happening here. For instance, in this point here, you probably have a lot of um, natural habitat, but it's definitely not delivering um, wild pollinators compared to here where you have a lot less, but you have a lot more wild pollinators. And this probably is, it would be useful to look more into detail, like which type of natural habitat do you have here and which type do you have here. And I think, for instance, over here you have a lot of coniferous forest, and here that has
Broad-leaved species, kind of.
yeah, and big patches of forest, so lot no a lot less edge vegetation, because we know forests, the edge vegetation also is very important, because you have shrubs, you have herbaceous species, you have the trees. While over here, for instance, I think you have less habitat, but it's more edge vegetation. It's probably all hedgerows, and tiny forest patches with a lot of edges, which provides both floral resources throughout the entire season, but also nesting resources. So it definitely would be useful to also, for instance, in this exercise, look at edge density, I think, and certainly habitat type, or to take out the coniferous forest here, for instance.
Yeah, I think it's very important because, like here in Rwanda, we do have like most of the woodlands are mainly composed of the eucalyptus tree species, and then the hedgerows, like for example, in the east parts of the country, we have most of the hedgerow are made of Euphorbia tricari, which is not do not having like some more the floristic plants in it. So it's very important again to include this component and characterize this semi-natural habitat and see what are the types of this and what are the hub layer of it. So things like that. Yeah. Thank you so much. Thank you.
Yeah. It also could be, for instance, the amount of crops that attract pollinators. It might be that, for instance, over here, the cherry orchard in which I sampled was the only cherry orchard, so all pollinators went there. While here you might have a lot more apple and cherry and pear orchards, so the pollinators are distributed. So it's the natural habitat is very important, but also the amount of crops that uh, demand pollinators or attract pollinators is important. Um, but yeah, it's definitely it's a short study of that I wrote, so it's good to read it because I try to explain it to the best of my possibilities. So this one, um, yeah, yeah. Thanks for the question.
Okay. Thank you.
Yeah, it seems like a good study. Maybe you can share the link to the chat room. In the
Ah,
chat
yeah.
room, please call now. Yeah, I would like to receive Fabrice Fatanya. He raised the hand.
Yeah, thank you very much for the presentation, dear Maxim. And I'd like to thank you very much for this interesting presentation as well. So, I've been collecting data on insect pollinator plant interactions here in Rwanda. It is in a master's thesis of a student called Tasia Hageniman. So yeah, I've been enjoying with the yeah scanning, of course, this interaction pollinator plants, and basically on some of the things you've mentioned on the pollinator dependence. Yeah, I would like to ask you whether you were planning or you did you had a look at pollinator dependence on the orchard you mentioned in your research. Uh, how how do you rate pollinator dependence on plants on
Yeah,
pollinating on plants as well?
yeah. Pollinator dependence is something we have measured extensively in blueberry. Um, and I also collected data of pollinator dependence of all blueberry studies across the world. Um, and yeah, this is under review at the moment. Um, and we see quite some differences of pollinator dependency across different cultivars of blueberries. So I think it's very important to measure this. Um, with Sean and maybe we also try to measure it for beans. And the way you measure it is with bags. So you have your plant, and before the flowers open, you put a mesh bag over the flowers so that the insects cannot visit the flowers and cannot pollinate them. Then on another plant or another branch on the same plant, you just label a branch and you count the flowers. And then if you have a lot of time, you can also choose a third plant or a third branch and manually pollinate another set of flowers. And in that way, you will have the yield or the pollination success, so the fruit set, for instance, or seed set of a branch that is bagged, so no pollination, only wind pollination. The second branch is then insect pollination, so that will probably have a higher fruit set or a higher seed set. And then you have the one that you also manually pollinated, so that will be a bit higher as well. And then the pollination limitation is the difference between the hand and the open pollination or the insect pollination. And the pollination surface, so the biotic value of the pollinators, is the open pollination minus the bagged pollination. I can send something about that as well.
Yeah, thank you very much. Uh, Let me see from the chat room what was written there. Uh, I can see some people expressing gratitude to Maxime for the great presentation. Then I can read also the comment from Gloria Kamwezi. She said, "Thank you for the interesting presentation. Quick question. How can farmers work with researchers or extension specialists to optimize with the pollination and honeybee management on their farms?" Can you respond to this first?
Yeah, oh, this is a good question, and what you need here is just a good collaboration with your farmers and do research in their fields so they can see what you do, and then also make sure you present the results um, every year. During my PhD, I had like 50 growers that I collaborated with, and every year I made a final report of the data we collected and what were the first results. Um, so that's one way to really have a thorough collaboration with them. But then, in addition to that, it's also very important to have a lot of informal conversations when you are doing the field work, when you're setting up the studies, to just talk about it and see if they what their questions are, what they think is interesting to research, and also sometimes um, incorporate these things to make sure that what you are studying is also relevant in their eyes. And I think these two things are very important in my experience. Um, and yeah, also to communicate, just to communicate. The main thing is to communicate. That's very important. The more face to face, the more important. And to be honest, if something doesn't work, then something doesn't work. But if, for instance, the the mason bees are very efficient, like this bee species here, we found that this is a very effective bee species. So now the last years, a lot of people are interested in how to manage these species. So they're gradually getting convinced that this bee species is useful. And now it's a matter of providing guidelines of how to manage this. Um, it takes a lot of time as well. If you
Yeah,
have a research program of 1-2 years, it's not possible, I think, because then in my opinion, it's also the PI of the group who leads the research needs to have that thorough network.
Yeah, thank you very much. Yeah, it needs a great work as I can see. And I think your studies can be replicated also in Rwanda. And if you talk to like Jaime and Kibogo and Fabrice, maybe you can establish some collaboration and look after some grants to do the same projects in Rwanda. That can be useful.
Mm-hmm.
I would like to welcome Kibogo Andrew.
Oh, yes, Venat. Thank you for this opportunity. I would also want to thank the presenter. It was a really very interesting talk, as I said early in the introduction. In fact, I just wanted to have a question. Actually, I saw in the presentation that Dr. Maxime was looking at different landscapes. I wanted just to know whether you are also looking at the actually at the abundance of the pollinators in these different landscapes of your interest. Uh, Actually, the other question, Vena has already mentioned it. I just wanted to welcome all the team, mainly those who are interested in the pollinators. We can embrace Dr. Maxime for future collaboration because I know here in the country, there's really a gap. We can do something. Thank you, Vena.
Yeah, I think it's an interesting step would be to just see which pollinators visit the different crops and also to measure the pollen limitation for multiple crops to just get an idea like which crops need pollination more than others, and also
Yeah.
like which species can then be possible candidates to manage or to support. Um, and regarding the habitat types, I did in other studies I did some surveys of well as well in other habitat types, and what you then see, and also in gardens. And then it became really clear that different habitat types support bees, but at different times in the season. Um, and this, for instance, I can also add a very interesting reference for that. Um, for instance, hedgerows with trees provide floral resources in spring, whereas grasslands don't provide these resources in spring, but the grasslands, they provide flowers in summer, whereas the hedgerows, they provide some flowers in summer, but less. So it's really, yeah, the complementarity of these resources is very important in the different habitat types. I think yeah, but if you are also for other people, if you have more questions, you can also put it on email if you want, and
Yeah,
we can discuss for sure. Um,
yeah, thank you. If
but
you drop your email in the chat room also, it will be great. Um, is there someone who has the last question? We would like to close the today's session soon, but I cannot end without looking if there is someone else who has a question or a comment. Yeah, maybe no one. I would like to thank you very much again, Dr. Maxime, for this great presentation. It seems like you have a lot of experience in pollination research, especially bees. Yeah, you are doing a great work.
thank
Also,
you.
Yeah, thank you all of you who attended the seminar today. Let us meet again next week on Wednesday, 3 p.m. as usual. The other thing, if you would like to speak to us to any topic, please get in touch with me and I will schedule you and you talk to us as well. We learn from each other, we grow together. Bye. See you next time.
Yep. Thanks for the invitation and good luck with all your research. And perhaps see you another time.
Yes, see you another
Thank
time.
you so much. Thank you.
Bye.
Bye.
Bye
See
bye.
ya.